morning, everyone, and thank you very much for the invitation to talk. Especially as all of you, I'm sure, know an awful lot more about viruses than you did 2 years ago, I would guess. What I'm hoping to do today is to fill in a bit of background, fill in a few gaps, and to get the chance to ask some questions. And this is just a brief overview of the virology of the illness that is COVID-19. The first thing to say is that there is nothing special about humans and virus, particularly COVID-19 and coronavirus. Viruses are everywhere. As far as we are aware, the viruses have been found in every living organism that we have looked at. They are ubiquitous in the natural world. They infect mammals. There's a seal virus that caused a bad epidemic a few years ago. Humans, obviously. All the animals that we know of have viruses. Elephants in zoos suffer severe viral infections that have a devastating effect on young elephants. There's a larch tree epidemic in the Lake District at the moment, which is killing trees. All plants have virus. One of the first viruses that I was taught about is tobacco mosaic virus, which infects tobacco plants. Insects have viruses. Algae, the fungi in the soil have viruses. Amoebae, plankton in the sea have been shown to be infected by huge numbers of viruses and present very pretty pictures when fluorescence dyeing is done on samples. So viruses are everywhere. They are the ultimate successful parasite that from genetic studies we know were around from the very, very early times of life developing on Earth. And there is even a school of thought that something like a virus was indeed the first sign of life development of a replicating being. So they are very, very successful, and they are successful parasites. And a parasite is an organism that has no capability of replicating itself on its own, but must use a host to replicate, and that's what viruses do. They are usually very species specific. They are very specialized, but as we know, evolution moves on so that changes take place. So what's happening here? We've got an idea of what the size of virus is, putting them in context with the rest of the world. On the right-hand side, we've got a frog egg that we can see, and then each of these pictures is 10 times smaller than the picture before. And viruses of all sorts usually come into the size area of about 30 to 300 nanometers. A nanometer is a billionth of a meter. The light microscope ends, not according to this schematic, but light microscope ends at bacteria. You have to have an electron microscope to see most viruses. Viruses need a genetic blueprint to replicate themselves. Our cells have a genetic instruction sheet of DNA, deoxyribose nucleic acid, a double-stranded DNA that we're very used to. And some of the viruses are in fact based on DNA, and they tend to be the more stable viruses that don't change very easily and are integrated very successfully inside their host cell. But the simpler blueprint genetic material is based on the single-stranded RNA, and this is where the coronaviruses come in. It is a simpler genome, but it's also more error-prone, so that some of its replications end in dead ends. They don't, they're not viable because mistakes have been made. But of course, making mistakes sometimes by chance gives the virus a new opportunity. And coronavirus and indeed influenza virus are based on RNA, and this is their basis for replicating and evolving. A parasite, as I've said, the schematic on the top left here is a basic DNA virus. What we've got in the middle is the genome. We've got a protein outer coat, the nuclear capsid, which protects the rather fragile nucleic acid of any sort, some sort of outer coating. And then the outermost layer is a capsid, a protein. In a coronavirus, it's a protein capsid. And then on the very outside, you get the mechanism, the little structures that are used to attach to the host cell where replication can take place. And coronavirus is an absolute standard virus for RNA on this model. We've got the single-stranded RNA in the center. We've got a ribonuclear capsid, which protects it, and then the outer capsid, and then the spike protein sticking out on the outside. Viruses are always made up of a modular structure, so that the absolute minimum of genetic information is required to make a new virion, a new capsid, and organism. So the material is the information for building, for instance, the outer capsid is very limited, and each of the pieces of structure in the outer capsid are identical. They're just like bricks building a house. You've got the standard single brick, and you can put it together to produce whatever you want. And that's exactly the model that is used for a virus. This is what a real coronavirus looks like in the big micrograph. This is an electron microscope picture. A scientist called June Almeida in 1967 was the first person to attempt to get a publication saying that she was a specialist working at the Common Cold Center down in Salisbury with David Tyrrell, looking at secretions that people produced when they had a cold. And this was a new one that she identified. It is said that her first paper that she put in to be published was bounced because the reviewers said, no, what you're doing is you're getting it mixed up with the flu virus, which is the picture in the insert. She went back and took more pictures, and she is the first published author of a paper identifying definitely a new virus, and she named it coronavirus because it looked like a crown on the top of the virus particle. We now have a lot of information about the genome of coronavirus. This is a schematic of the coronavirus genome. It's 30,000 nucleotides long. This is it stretched out. Part of it we know, I should say this sequence of the original virus that was identified in Wuhan was released to the public in early January 2020 by a Chinese scientist. So this sequence has been known for almost the whole length of the time that the infections have been going around the world. This area, ORF, is open reading frame, very loosely equivalent to our genes in our genome. This is the area that produces the structure, the protein structures of the virus. This area produces the messenger RNA, which is basically the instruction sheet for the virus replication. What we're going to hear a lot more about later on is the material produced by this area, and you won't be surprised to hear that it's the spike protein area. And this part of the genome codes for the structure, the protein structure of the spike that is instrumental in getting the virus particle attached to a relevant host cell. As I've said, this virus, this RNA usually can be quite sloppy in how it replicates itself. Sometimes that can produce useful changes in the spike protein area. If you make a mistake in this area, of course, the virus is going to be a non-viable particle. If a mistake is made in this area, it may present new opportunities for the virus. So that is how new viruses are made, RNA viruses in the main. These mutations, or variants, as we're now calling some of them, give evolutionary opportunities. And we know this from many instances in the virological world. One very good example is measles virus is very closely related to distemper in dogs, and is very closely related to rinderpest in cattle. Now, none of us can catch rinderpest, and cattle can't catch measles. But we know those three viruses are very similar in their genomic situation. They're very close cousins, and they had a common ancestor not too long ago, probably just after they must have probably split up just after man started keeping dogs and raising cattle. So it's a very good example of how a common ancestor has taken advantage of new opportunities and has changed slightly and been able to, by mutation, new opportunities of new hosts. Flu does this by a reassortment of genetic materials, again, close contact with chickens and ducks and pigs. So other RNA viruses give us a very good idea of the potential for this. As I've said, it's the mistakes that give the opportunity, but there is a downside that sometimes a lot of non-viable virus particles are produced. But occasionally, a new opportunity is found by chance. And what's happened with CoV-2, the virus, it has mutated, it has jumped, and the result has been a completely new virus in a completely susceptible population, and that is a pandemic. And it continues to evolve. And the mutations in the spike protein is where the new variants turn up, and that's what David's going to be talking about in more detail later on. This is a nice simple schematic from the virus's point of view of what happens in a cell. The virus with its in this case, coronavirus, has a spike on the outside, and it's got to find a receptor, a molecule on the surface of a potential host cell that it can attach to. And it's as tight and as specialized as a lock and key. And those two have to come in contact with each other. When they do, the virus attaches to the outside of the cell. It then makes entry into the cell by a variety of different means, either absorption through or by active bubble vesicle being drawn into the cell cytoplasm. Once the virus is inside a cell, it uncoats itself. The capsid disintegrates and the genetic material is released. Once that genetic material is released, replication then starts. And all the components of new, not identical, but mostly identical virions are produced inside the cell. And then eventually those individual structures are assembled together to make a new virus particle. This schematic shows the outer surface budding out of the membrane. That happens with flu and some others, but it doesn't happen with coronavirus. It has a different method. So this is a nice simple way of looking at replication from the virus point of view. And it's successful. It happens within 12 to 24 hours within a susceptible cell, and thousands of new particles are produced. But of course, a host cell is not an empty bag. Molecular cells are very, mammalian cells and all other cells are very complicated. The cell membrane is where there has to be a receptor for the virus to attach to. The virus has to get into the, past the cell membrane, but cell membranes are very dynamic. They have material coming in all the time. They have waste products going out. There's energy diffusion. And one of the ways that molecules are brought into the cell is a pinocytic vesicle. A little bubble is drawn into the cytoplasm. This is the area that the virus, coronavirus, takes over. This is the assembly line in any living cell. The nucleus, of course, is where the brains of a cell is, but the actual manufacturing process takes place in the endoplasmic reticulum. And this is where the virus sends new instructions for its own structures to be produced. And it will then completely disrupt the normal working of a cell and take over the manufacturing process. In the case of coronavirus, CoV-2, we know that receptor on the outside of the cell membrane is called ACE2. It's present in a lot of cells, most primarily in the lungs, which we'll come back to. That is the receptor that the spike protein has to come in contact with. We now know that coronavirus doesn't bud out of the cell or burst the cell open to release virus particles, but in some cases, it slowly disintegrates the cell membrane and joins or goes into the adjoining cell, so that in the end, you end, in a certain length of time, you get a massive multi-cell. They're called syncytia. And this may well be a method to evade being noticed by the immune system, which we'll come back to. But eventually, because it's cell-to-cell passing of virus particles and a way of infecting a new cell. But in the end, huge numbers of viruses will be present, and then they release themselves. And that's it as far as the virus is concerned. A successful infection of a host cell, reproduction, release, ready to infect a new cell. And that's in a very short time, 24 hours, 48 hours for any individual cell. And thousands of new particles are produced. So that's the virus's story. For the host, it's different. Before I go on to COVID-19, I have to mention the common cold. I'm not sure if you can see it on my screen. It's covered up. But this is a common cold, an upper respiratory virus infection that routinely, most of the viruses that cause the common cold infect through the nose. They replicate in the sinuses and the back of the throat, cause a lot of damage, and we all know the symptoms of that. And viruses have found this a very useful place to replicate themselves. And there are over 200 different strains of virus that use this particular set of cells to replicate themselves. And because there are over 200 different strains is one of the reasons why we get cold after cold. And there are 4 strains of coronavirus which cause that. And that's where the family was first identified from, as I've said. And of course, children are extremely good super spreaders of cold viruses. They have quite poor nose and hand and finger hygiene. They splutter all over each other. And of course, they've not come in contact with any of these viruses before. So they're very good at spreading the cold virus. Flu and COVID have some similarities. The main similarity being that they are both infected. Those infections are transmitted via the respiratory route. And we can say that influenza is a respiratory virus. COVID-19 is not a respiratory virus. It's transmitted by the respiratory route, but it is a systemic virus. Transmission, as we now know, droplets and aerosols from the nose and mouth spread the virus. When it happens when people sneeze and cough indoors, there's significant transmission. Outside, not so much. We were very worried about survival on surfaces at first, but real world data does not suggest that this transmission by surface containing virus is very, very unlikely. It's inactivated. Viruses in general, but coronavirus in particular, is inactivated by alcohol. That's the sanitizer. But very importantly, viruses are inactivated by chlorine and by UV light, and that is the whole basis of our clean, wholesome drinking water system to get rid of the viruses. I'm not going to say anything about symptoms because you all know the symptoms. The only thing I will say, the symptoms come into 2 categories. Something like a cough is the direct damage to the lungs and throat by the virus destroying those cells in the body. The other reason we have symptoms is because of the reaction of our immune system to that foreign particle and the damage that it produces. And I'll be saying a few words about that in a minute. The lungs are the primary route of infection, of transmission, down and down into smaller and smaller tubules until the air sacs at the end of the lungs, the alveoli, are lined, as this microscope picture shows. It's been stained. These are ciliated columnar epithelial cells. They line the alveoli. They have cilia along the top there. to waft along the mucus that is produced by these goblet cells that are stationed all the way along this line of cells. The mucus is there to be moved along to get rid of debris that we breathe in, pollen, dust, particles in the natural situation, and also to move away the dead and damaged cells and old cells that are in this layer as well. As far as the virus is concerned, anything that is breathed in by chance, completely randomly and passively, will find themselves in this mucus stream. It's quite a good transport system, but it's also a barrier to these cells, and it is on this cell surface that the ACE2 receptor is found, and that is the area of the cell surface that the virus must come in contact with to initiate an infection. And ACE2 molecule is found all over these epithelial cells. The virus then initiates an infection, and we then have a response. We have a system that responds to any foreign material or local damage, and it's called the innate immune response. Fever is part of it. That's a way of energizing all our immune system. And there are two other main areas. We have the white blood cells, the phagocytes and lymphocytes. They're activated to go and target any foreign material, any material that is not self, but also to clean up local damage. Old cells have to be removed. They are the doers, if you like, the cytokines in the humoral response down here. These are small molecules of proteins. The complement cascade is an example of one of the cytokine systems, and they are the instructors. They are the molecules that that organize and regulate what happens in the immune response. This happens no matter what the foreign material is, and the immune system is very good at recognizing non-self, but it is very non-targeted, non-specific, but it is immediate. What is not immediate is the innate immune response. And this is a more sophisticated, targeted response by the immune system against a specific foreign particle. And our coronavirus obviously comes into this, but it takes time. But the time, 10 to 14 days, is when the targeted lymphocytes are produced, a specialized white blood cell. They're produced, and they target specific areas of, in this instance, the coronavirus. And again, the humoral response, one of the aspects that happens is that the B cell lymphocytes produce specific antibodies. You've all heard the word antibodies a lot, I'm sure, even if you didn't know of them before. So the acquired immune response takes time, but it means that there is a memory of this infection which can be used in the future. And of course, vaccination is our unnatural, our artificial way of producing this same acquired immune response. This is a picture I've taken out of a textbook. I'm not going to go through it in detail, but what I liked about it, it gave, I mean, this is us being infected from all different angles, and the humoral, and this is the acquired immune response. The humoral response is the B cells are activated to produce and secrete antibodies, and then the antibodies attach to the virus particles. But we also have the cell-mediated response, where the T cell lymphocytes are produced and they kill cells and they produce other cytokines. But there is a big feedback loop here, and this is regulation and activation is very complex, and it's this that sometimes can go wrong and cause us problems. COVID-19, the severity of the infection we know an awful lot more about nowadays. There are a lot of asymptomatic infections, but severity, the disease, the infection can result in very severe disease, and we know death as well. Age and underlying health are really important. Genetics, ethnic group, initial dose, all make a difference. Viral pneumonia is direct damage by the virus that causes so much damage to the lungs that they stop working well. If that happens, the bacteria that lurk in our lungs take advantage, and they can cause a secondary bacterial infection. But also, our immune system can, in some cases, not be well regulated, and it gets out of control. We know that the ACE2 receptor is found on blood vessel lining in all sorts of different organs. So the virus can cause inflammation and targeting by the immune response in all sorts of organs throughout the body. And the worst case scenario is a completely out of control immune system called a cytokine storm. And that's one of the reasons that death occurs. Testing, PCR, polymerase chain reaction, very sensitive way of finding out if there is a current infection. The downside is that it will also pick up the remnants of a past infection for a little while as well. The lateral flow test, which seems to be on every street corner available now, it's not as sensitive as the PCR, but it does pick up the spike protein and is able to give a very quick result. PCR has to be done in a laboratory, and on a good day it will take 24 hours because there's some astonishing laboratory setups out there now. The lateral flow we know within half an hour or so. Also very important is the antibody testing for past infection, and antibodies are important, are the way we find this. After a natural infection, two sorts of antibodies are detectable. There'll be a lot more others produced, but we can detect the anti-nuclear capsid antibody and the anti-spike antibody. And this will show that it was a natural infection. After vaccination, only the anti-spike protein is produced. And there are lateral flow tests using finger prick samples. Real world surveillance has is bewildering at the moment. There are so many different studies out there. Almost every day we get a slightly different view of the case numbers, the deaths, the antibody levels, the age group possibilities. So I'm not going to say any more about this. It would take another lecture to do that. I will just note the ones, being an ex-laboratory worker, I like being part of a laboratory project, a research exercise. So I am a very dedicated Zoe tracker. And in fact, Zoe made the news today. They just reported on looking at COVID infections in children and come to the very good conclusion that serious illness is very rare indeed. I'm also part of Biobank, have been for 10 years or so, but they've been doing a lot of active testing at the moment. And in fact, I'm waiting for an antibody test result for them to see how long-term my antibodies are. I'm also interested in the DESPRA sewage monitoring. COVID is shed from fecal material, and my particular research interest was viruses in water and sewage. So I'm taking a particular interest in that. But all of these, there's a huge number of scientists doing a huge amount of work, and it can be quite confusing, their results. But big pictures are coming out. Lastly, I have to mention vaccines. I'm not going to mention anything else about vaccines because, again, that's a completely different talk. But as I've said, they are the artificial way of priming us against future infection. Again, there's huge numbers of detailed studies coming out, but I think the general picture is that if we are fully vaccinated, we have a very small risk, not a nil risk, but a small risk, very small risk of getting reinfection. That infection is likely to be fairly mild, and household contact studies have indicated that transmission within households is very low for fully vaccinated people. But the virus is endemic now. It's not a pandemic anymore. It's endemic in our community, and we have to cope and learn to manage it, just like we have all the other viruses that have successfully used us as hosts over the millennium. And that's it. I will stop sharing.
Brilliant, brilliant. Thank you very much indeed, Jane. Very well illustrated, and an awful lot of information contained in those slides. It's staggering what we know about the inner workings, isn't it, of the virus and cells in general at such a small scale. I mean, we're talking sublight wave length, aren't we? And electron microscope type length. Okay, let's have a look at the chat box. There are one or two questions. From Mike Warren, is that human DNA? I think he was referring to one of your early slides where you showed the double helix and the single helix.
Yeah. Most of our mammalians, most of our cells, all of our cells are basically, the genome is double-stranded DNA. It only splits when in the reproductive cells and things. So yeah, we're made of DNA. We have a lot of RNA. That is how the DNA sends instructions to each of our cells, within each of our cells, and the RNAs are the workhorses. They give the instructions to the for the workings of each cell.
Okay. Another question came to me. I don't think you answered it in your talk. It's from Alex from Weymouth. He says the furin cleavage site the Chinese inserted, how does this make the virus more virulent?
I'm not sure I agree with the first part of that. That's getting on to, is this an artificial virus or a natural evolution? And that's not it's not something that I would be able to comment on, but I don't personally, from what I have read from virologists and heard, I think this is extremely likely to be a natural infection evolving, as so many viruses have in the past, for instance, SARS. And I think that's as much as I can say on the subject.
Yes, okay, Jane, thank you. Peter Weston, what sort of research is carried out in Wuhan? Is it possible that it could facilitate the mutation from bats to humans?
That's the only comment the only comment I will make is Wuhan Virological Institute was set up after the SARS outbreak. It came before. It was doing virus testing, virus research, because the SARS virus was so very deadly and was a real potential hazard of a severe pandemic that we needed to have good scientific research.
Yes, yes. Mike Warren says, does the efficacy of the vaccine decrease over time? He says some reports say 3 to 5% per month. Is there a...
Not that I'm aware, no. That you get to a certain state, the vaccine takes 10 to 14 days the vaccine contains information to produce the spike protein. That spike protein is then present in your muscle cells, and the immune system recognizes it and starts to produce a response to it. It takes time for that response to be optimized, and it's certainly the case that within 9 or 10 days the vaccine gives you no protection at all. After 10 days, I mean, we always used to say for rubella vaccine, measles vaccine, 10 to 14 days, and then it was okay. The public health officials are being much more cautious nowadays. They're saying 21 days is the time where there's the maximum amount of antibody detectable. You have to remember that where detectable antibody is just one way that we can actually see what's going on. There's a lot of other things going on in the immune system that are not easy to detect. As far as there were early studies of people getting infected after vaccine, and certainly from 21 days should be about the optimum time. What we don't know is how long that optimum immunity lasts, and certainly the second dose boosts it. It really does seem to make a big difference, and again we get this jump in the amount of antibody, and we know from real world studies that people are protected much better after a second dose. They are saying after 14 to 21 days, which surprised me, because the immune system is very good at really getting going if it's got a memory. So, but again, they're being cautious. So 3 weeks after the second dose is the optimum amount of immune response. We don't know how long that response lasts, and we won't know until time passes. But we do know detectable antibody wanes, but how that relates to the ability to fight a new infection, we will have to do lots more studies to find out.
Okay, Jane, thank you ever so much. I'm getting a handful of more questions in the chat box, but time is moving against us. So
Steve, can I interrupt? Steve, can I interrupt? I think David McLellan wants to make a comment in response to the last one. So
okay, go on, David.
Yeah, just a comment about the longevity. I think one of the problems with when we talk about antibodies is many of them are transient. The most important thing is how we respond the next time round, as was just said. And it's those memory cells that are so important. I think one clue to this is the original SARS virus. People who actually recovered from it, that's 20 years ago, we can still find immune cells in their bodies which will respond to that virus.
Wow.
So, I mean, that gives you a clue. But as Jane says, we will not know until things sort of evolve time-wise.
That's really interesting. Okay, thank you again, Jane. that's really great. We'll pick up the other questions, I think, when David's finished, and we'll pick up all the questions as far as we can in the time available. So please take it away, David. Thank you very much.
Try and share screen.
Yep, that's working.
Yep. Okay, well, that's me. I'm from Torridge U3A, and as you rightly say, I'm going to be talking more about the variants and things associated with them. So, just a reminder about the terminology, because we tend to use these terms a bit loosely. The virus is SARS-CoV-2. The actual condition that it causes is COVID-19. So we need to be aware when we talk about COVID that there is these distinctions. And of course, this is an example of a figure of what the virus looks like, as we've been shown earlier. And you can see the little spikes on the surface of the virus, which actually are made up of 3 identical components. And it's this area here that we're going to be mainly focusing on when we talk about the mutation. Now, a namesake of mine in America produced this rather lovely 3-dimensional figure. Now, this is one of the, we can actually only visualize from here, one of the spike components, but it's the surface here, which is where the binding of the spike protein to its receptor occurs. It has 2 main components, 2 main sections. It has a recognition component, and that's the top half, this bit here. And it actually then allows it to, as was said earlier, as Jane said earlier, it can actually attach then to ACE2, which is in full angiotensin converting enzyme. And these are on a range of host cells. And then below that, this area at the bottom is what's referred to as the penetration component. And somebody mentioned furin cleavage. Well, cleavage has to occur before the virus can actually penetrate the cell. And this happens in a number of ways, but there are 2 cleavage sites in this sort of area which enable that process to occur. Just briefly, a consideration of what ACE2 actually does and why it could be important in terms of how the disease develops when you get the virus. It's vital for quite a variety of biochemical pathways, which I won't go into detail, but particularly it regulates the following. Blood pressure is very important for. It has a role in inflammation, and if you'll mostly be aware, inflammation is a major factor when it comes to disease progression. And also in wound healing. These are 3 processes which it's involved in. The other thing is it's present on the surface of many cell types. For example, it's found in the nose and mouth and in a wide variety of organs, as was mentioned earlier. And although we tend to focus the attention on the lungs and the trachea, etc., have to remember that there are receptors in all of these organs, on the tissues of all of these organs, the heart, liver, kidneys, the gastrointestinal tract, blood vessels, and to a limited extent, the brain. We've already seen a figure, this is slightly enlarged, but this is the figure of the COVID virus actually getting into the cell. It attaches to the cell, as we've heard, it has to be cleaved, then it enters the cell, replicates. But the one thing it also does is it actually affects the normal function of the cell that it's actually penetrating. So it's not just damaging, it's not damaging the cell, it's actually damaging the function of that cell. So really, what do we mean by, just before we do that, it's worth just mentioning that the original SARS virus, which arose in 2002, which I mentioned earlier, disappeared in 2004. We might hope that the same thing might happen for this new parachute. It was, as was said, far more lethal than COVID-2, but less communicable. And that's an interesting point. And that's partly because it was less efficient at binding to its target cells. Unlike COVID-2, where a third of those infected are symptomless, COVID-1 appears to have been spread by subjects who showed respiratory symptoms, and so it was therefore somewhat easier to contain. Okay, we can move on. So what do we mean by SARS-CoV-2 variants? Well, as was mentioned earlier, all viruses change, but some change more than others. And this RNA virus seems less variable than many viruses, including flu. I think it's something like I'll be corrected in this, I'm sure, 4 times less likely to change than the flu virus. But it's far more variable than certain other viruses like measles. Critically, and I think this is quite amazing, over 12,000 mutations in the virus have occurred since it was first identified in China in the early part of, the late part of 2019. And the term variant has been used rather variously in terms of, is it a single point mutation or is it a whole range of them? But generally it's used to describe a virus that's acquired several mutations. In other words, a cluster. And this is what we're more familiar with when we talk about the key variants. Now, the terminology for these viruses and their mutations has been quite complex, and it has been simplified to some extent in recent times so that not just scientists, but the lay person can understand it a bit better. Back in the end of 2020, as some of these variants started to emerge, the WHO, World Health Organization, with a variety of other networks, researchers, etc., started to group the variants into what they considered to be 2 main categories. Variants of concern and variants of interest. We'll go on to the variants of interest a bit later, but variants of concern is the main one. It's interesting to know that in the UK, these are referred to as variants under investigation. And I suppose you could say that's slightly more scientific than variants of interest. I mean, we might be interested in it, but in fact, it's the investigation that is important. The Americans have to be slightly different, and they've actually got a 3rd category, which is where there is total escape. But fortunately, none of the variants have actually got to that point now where we can't develop immunity to it, or it causes really severe disease like COVID-1. So what do we mean by concern? What did the experts decide was concern? Well, this is where the mutations can lead either to an increase in the viral transmission, or it can lead to an increase in how severe it is, and medically, in other words, its virulence, or some other changes in its clinical presentation. Or where there's a decrease in public health measures, for example, diagnosis, and also where you've got some abnormality in terms of response to vaccines or other therapeutics. But currently, we still have only 4 variants that come within this category. When I gave this talk in its earlier form, earlier in the year to our local group, there were only 3 variants identified as variants of concern. And so we've now got the 4th one, which came out of India, but we're not supposed to say that now. And the same way we're not supposed to say that the labs in Wuhan caused the virus to become human orientated. And the WHO named these 4, gave these 4 variants alphabetical numbers. So we have an alpha mutation, which is originally the Kent or UK version. We have a beta, which is emanated in South Africa, a gamma, which arose in Brazil, but I think was initially detected in some Japanese individuals who'd actually been traveling there. And then, of course, we've got our latest version, which is called Delta. And this one emanated originally from India. Now, the country region was the thing that the WHO were concerned about, because he thought it was a bit of a slur, if you like, on the country that it was named after. Whereas, in fact, it was very often the ability of our scientists to detect this, which enabled us to say that it was actually present in the UK to start with. There are other names given to these viruses I'm not going to go into, but these you'll see as I talk about each of the individual COVID variants in turn. Let's start off by looking at the alpha, or as was known, the Kent UK variant. Now, you can see what I mean when I talk about a cluster, because this version had at least 17 viral protein affecting mutations. Now, as was said earlier, the spike protein is the key to this. It may not be the only factor, but it is a key factor. So 8 of these mutations affect the spike protein of the SARS-CoV-2 virus. Now, one of these, which is referred to as N501Y, which seems very obtuse, many years ago, amino acids were given a simple code, an alphabetical code again. And so this refers to the 501st amino acid in the sequence of the spike protein. And it just shows you that there has been a mutation which has actually affected that particular site, and it's actually changed the amino acid at that site. I won't go into further detail about that. But because it was affecting the S1 component, you'll remember that's the bit that really comes into contact with the receptor. It was referred to as the S1 variant. It's worth mentioning at this point that the docking area on that S1 component is usually referred to as the receptor binding domain, and many of the variants we're going to be talking about can have an impact on that particular vital domain which interacts with the receptor on the human cell. This mutation actually increases the capacity of the virus to bind, and that's a very important factor. It could have gone the other way. It could have created a mutation which reduced the capacity to bind, which would mean it was less infected. But this one increased the capacity, and therefore it infects its human target cells more easily. It increases the rate of transmission, and that was obviously very important because it increased it by something like 30% greater. And there were some initial concerns that it actually might affect the virulence, and this came out from about 3 different reports from different universities, all giving different figures and different values. And in hindsight, it didn't seem to have a significantly increased rate. The most important thing was the rate of transmission. And it can increase the viral load in individuals. Obviously, if you can increase the viral load, you obviously can increase the number of particles that are going to be expelled, etc., when someone coughs, sneezes, etc. Now, this is a thing that's emerged on the top of my. Unmute my. Can you still hear me, folks?
Yeah, go ahead, David. That's fine.
Okay. Yes, it came up to say I was mute. Okay. So one very important factor, which we must be really proud of, is that we've got one of the major viral genomic hubs in the world. This is a hub of various contributors across the UK, which look at the genome of viruses, and particularly this virus. And they were the ones who picked up this variant initially in a cluster of cases in Kent. That's how it got its name, Kent. It seems to have emerged in late September, and you'll notice that when we look at the key variants that we're going to talk about, or we're talking about, that they all emerged in a very short time span, all between about September and December of 2020. By mid-November, it had comprised something like 30% of cases in London. Now, it's worth putting this into context because prior to that, there had been another variant. It wasn't called a variant then. A variant which allegedly came out of Spain. And that was a particularly spreadable virus. And in fact, most of Europe, and particularly the UK, was the Spanish variant at that time, but it was soon replaced by this alpha variant. And after 2 or 3 months, it became the key variant in this country. And you can see how quickly it took over from the original key variant. The blue bars illustrate what I would call the Spanish variant, and then the orange bars show the increase of the Kent and London variants, so that from the beginning of October to the end of December or middle of December, you can say it's virtually taken over as the variant of concern. We then got slightly worried about another variant. Information was coming out from, I wish I could get rid of this thing with the sign. I don't know why it's doing it. Anyway, they reported a new coronavirus variant, and it has a comparable number of mutations to the UK variant. Only one of them is the same as the UK version. So this is this is the mutation that enhances transmission and infectivity. But there was a concerning additional mutation which had the potential to reduce antibody recognition. Now, obviously, if it does that, it's going to be a big problem for vaccines, let alone our own individual responses to the disease. However, after initial concerns, this mutation has to date had little impact in the UK. It didn't penetrate in the same way as the Kent variant, and it hasn't become the dominant variant in this country, as we'll see later. You've read, and some of you will have been affected by the fact that it's present in parts of Europe in various amounts, but they are relatively small amounts. There was some concern it was actually taking off a bit more in France, but it seems to be coming under control, or it seems to be being overtaken by a new, more adaptable variant. And this is the thing, it's the adaptation. If that wasn't enough, this Brazilian variant, which was originally known as P1, emerged in, again, mid to late December, so to the latter part of last year. As I mentioned earlier, detected in 4 people traveling to Japan. And again, it shares several of the mutations, particularly ones on the spike of the virus, and 2 of which are seen in the UK variant. The new Brazilian variant is also associated with an increase in infectivity and the potential, and this was of concern, reduced antibody recognition. And they started to pick up cases of people who had developed immunity and have then got another dose of virus. We're seeing that with the latest variant in this country. Again, this variant had little impact in the UK, and I'm not saying it isn't of concern, but it's certainly of less concern in this country now. The one that's replaced all of these, and we're all now familiar with, was Delta (Indian variant. This was recognized fairly early on, but it wasn't recognized as a variant of concern until May 2021. Now, you may ask why there was such a long gap between when it was first noted and when it became a variant. Obviously, there needs to be observation as to whether this is a transient virus. The other thing is as to what its penetration is in populations, and whether it's actually overtaking other viruses in terms of becoming the dominant virus. And it was soon becoming apparent that it had very high transmissibility. It's a little bit more complex because 3 subtypes have been described of this named virus. And you can see in red 2, which illustrates it's the second of the 3 subtypes. And it's become dominant in many countries around the world, and it makes up most, if not all, of our virus now. It has, again, several key spike protein mutations. 2 of these affect the ability of the virus to bind the target. It actually improves it. And others may allow evasion from host immunity, which is always a concern. It's far more transmissible than the alpha variant. And if we go back and try and compare it to the original Wuhan variant, the first one that appeared, it's probably 2 to 3 times more transmissible than that, which is very important. And again, has the potential to reduce antibody recognition. Of interest, laboratory studies, which we refer to as in vitro rather than in vivo life studies, indicate it may be up to twice the invading capability for lung and colon cells in human beings, which matches this idea that in fact it's at least twice as transmissible as the first native virus. However, after all the initial scare and concern and headlines in the newspapers and from certain politicians, it doesn't appear significantly able to reduce the efficiency of the current COVID vaccines, particularly the so-called double jab community. So if people haven't had their jabs, they should go out and get it, because all those who are going into hospital and most of those who are dying, as shown in the States, where it's taking off there in a large manner, are people who haven't been jabbed. So if they haven't been jabbed, they stand a much higher chance of getting serious complications, particularly if they have some of those conditions which are inflammatory, like obesity, diabetes, etc. Now, we dealt briefly with the variants of concern. Also ought to mention something about the variants of interest. There are currently something like 9 listed variants of interest, or as we call it in the UK, variants under investigation. And these include, in order of the date when they were designated, and don't ask me why they're not in order in terms of the alphabet, the following: Zeta, seems strange that should be in the order of designation, Eta, Theta, Kappa, Lambda, which I've underlined in and placed in red, Epsilon, Iota, I'll go back actually, and Iota. Now, it's interesting that I'll just concentrate a little bit on Iota and Epsilon. These emerged in the United States, in California, earlier last year, and were initially considered by the Americans, and bear in mind it was the Americans, they didn't feature in terms of the WHO, as variants of concern. However, scientific evidence suggested that in fact the numbers were significantly decreasing as far as variants circulating in the United States, and also they got some data which indicated that vaccines and treatments were effective against the variant. So what they did was to de-escalate the variants, these variants of concern, as they call them, to variants of interest. So these are fairly in that area. Now, what we don't know is if any of these variants of interest could emerge as variants of concern. In other words, could they take over, for example, from the Delta Indian variant? There is one which does seem to be of some concern, and that is Lambda. There is some indications that it has some similarities in terms of its transmissibility to the Delta variant, and therefore could potentially emerge as a variant of concern. But that's all speculation at the moment. I think what's interesting in terms of emergence is that all the 4 variants of concern emerged at the end of last year. We haven't had any variants of concern that have emerged in the last couple of months, 2 to 3 months. Doesn't mean to say we won't, but it is interesting how they emerged at a very similar time. One thing which is puzzling people is how did these new variants arise? Well, we all know that they happen spontaneously, but there are certain things that may escalate that process. One possible cause is that the individuals have a chronic infection with COVID. And it's speculated that this situation can occur in particular individuals with a weak immune system. In fact, there was long-term studies showing 2 or 3 patients who didn't deal with their COVID infection well because they were immune compromised. And in that period of time, they were able to pick up different mutations occurring to the virus in that one individual. They could then pass that multiply mutated virus onto other people. persistence of the infection would enable the virus to evolve, and it might explain the large number of mutations in these viral variants. It's not the only cause, and you'll often see it stated that vaccination is going to put pressure on the virus and it will mutate for that reason. On the other hand, people will say if there's lots of the virus circulating and replicating, that's the main cause of mutation. Mutation will occur regardless, and I don't think we need to worry necessarily that being vaccinated means that we're going to be producing huge numbers of variants. Well, we've already indicated that these new variants don't appear to be evading current vaccines. And it's important to consider that the antibody responses to the spike protein, which is has a large number of components to it, comprises really a soup of different types of antibody to the S1 component, this critical component. Various studies, both here and in the United States, have shown that the main vaccines, as I indicated earlier, are performing appropriately, and it's been suggested that they reduce the chances of hospitalization or death by as much as over 90%. However, we must be aware that other novel mutations could occur, and they obviously could possibly evade the current vaccines. This may be overcome by modifying the vaccine to enable the virus variants to be neutralized, and we now have the methodologies in hand that we can do this fairly quickly, particularly with the RNA vaccines. And this just illustrates how a vaccine, or actually say a variant, could escape a vaccine. Here we've got 3 different antibodies. These Y-shaped blue shapes here are 3 different specificities of antibody. And they bind different sites. This is a slightly rounded site, this is a slightly squarer site, and this is a sort of triangular site. So they're all seeing something slightly different. Now you can see what could happen. You could have a mutation which would stop, and this is the mutation that's occurred, where it's gone from being round to a square, stops that antibody from recognizing that particular site. However, it's still got these other 2 friends who can actually still work. Recent studies indicate that protective antibody immunity will be long-lasting. We were talking about this earlier, via the generation of memory cells responsible for antibody production. And there's a lot of work in it now which is illustrating this. And another very recent study has shown that highly potent, wide-ranging antibodies, in other words, against a whole range of different specificities, not only for the spike protein, but for other proteins on the surface of the virus, can actually be identified in people who've actually been inoculated deliberately with SARS-CoV-2 variant, or a particular variant. These antibodies were capable of not only neutralizing variants of interest, but also some of the variants of concern. So it does suggest that our immune system is very adaptable. Some of the anxiety regarding antibody evasion actually relates to a particular type of immunological therapy. Now that antibody there, if it was the only one being produced, could be referred to as a monoclonal antibody. It will only pick up that particular site. This is a polyclonal response because there are 3 different antibodies looking at the protein. This one's a monoclonal response. Now you can imagine if you're treating somebody, as this has happened, and it happened to Donald Trump, you're treating them with monoclonal antibodies, there could be evasion and it could be quite serious. But there are a whole range of monoclonal antibodies that have now been lab produced, which if one doesn't work, others do. Right, now finally, we now have the potential, and as you heard earlier, we're extending it to the older teenagers, of actually getting people vaccinated by September. We can, as was indicated earlier, anticipate periodic resurgence of the virus, just the same way as flu does. And as was said earlier, what started out as an epidemic in China and became a worldwide pandemic is now believed to be endemic. Like flu, we're probably going to have to be jabbed, certainly in the foreseeable future, to protect the vulnerable in winter. With the new vaccine production techniques, and well, I've used this nice modern term, I don't know what it means, toolbox, and treatments techniques improving as we speak, it should be possible to react very quickly and be able to produce suitable vaccines where they're needed. One last thing before I go is that the toolbox now contains not only the novel RNA vaccines, but the prospect of novel plant-based vaccine technology. At least 2 companies are in the clinical trial phase of production, including a company from Canada called Medicargo and the British-American tobacco firm. Goes back to the tobacco plant again. Medicargo is using plants as bioreactors to produce what they refer to as virus-like particles. They don't have any infective agent, but they have a shell-like structure similar to the virus, which is studied with all the antigens required to trigger immunity. They're into clinical trial phase. I think they're into trial 3, and which is a sort of later stage before it starts to get rolled out if approved. Early studies indicate a very strong antibody response. Now, antibodies, as was indicated by Jane, aren't the only part of the immune response, and we do tend to forget how important it is that other cells, other immune cells like T cells, are involved. They have a very important role. If approved, these vaccine products could make serious inroads into current worldwide vaccine deficit. I think it's been estimated that something like 25% of the Western world has been immunized, whereas something like 1 to 2% of the rest have. Medicargo predict they could produce up to 1 billion doses of vaccine by 2023. So we just wait and see whether that happens, whether it's fast, and what transpires. But I think the future is bright. Thank you very much.
Thank you, David. Absolutely brilliant, very instructive, and you've added a lot of clarity to our situation there. Very much indeed. I'm looking at the chat box, and of course time is running against us a little bit, but one comment, well, I've got a couple of comments that allude to the lessening of importance given in the press about surface contamination and cleaning, and you know, how careful should we be when touching surfaces or receiving deliveries at the doorstep and so on and so forth? Is that so much of a concern generally? Do you, either of you think?
I think Jane mentioned this earlier, so go get back to Jane about it.
Yeah, good hand hygiene is always a good idea. First of all, I believe CDC have now removed the need for detailed cleansing of surfaces as a way of dealing with the epidemic, with the infection. There doesn't seem to be any strong evidence of transmission that can be pinpointed to surfaces. But it has to be an individual person's risk assessment. But I think it's very, very much less than person-to-person contact in the same room.
Okay, Jane, thank you. Barry, would you like to sort of pick up one or two questions from the chat box? And we'll need to keep an eye on the time, I realise that.
Yes, I'm just looking through it. Someone's asked a question here. Mike Warren has asked a question. Well, what is actually the meaning of viral load, and is it related to these so-called super spreader events?
No, viral load just means the quantity of virus that is present at an infection. So yeah, I mean, a super spreader would be carrying a lot more virus, but it's the term we use for the amount of virus that goes in at any one time. And as you can imagine, in aerosols and droplets, viruses don't travel alone. They are stuck together, and they have clumps, if you like, of viruses. And the more virus you get in one episode, the more likely it is probable, the more likely you are to set up an infection. So it's just the quantity of virus present.
Okay, thanks, Jane. David, yes, you've got a comment on that.
Just to add to that, I think that there are other factors involved as well, and that is the number of the receptors that are present on the surface of cells. And as we know, kids, very small children, don't actually suffer clinically anywhere near as much as adults. Partly this is down to the fact they have fewer ACE2 receptors on many of their cells. And as we age, we tend to produce more. And if we're male, we produce more. And if we've got inflammatory conditions, we produce more. So the viral load obviously would have an influence there, because the virus gets in, and the more receptors there are, the more those cells can then get infected and produce more virus. So there were the various aspects.
Thanks both of you. So Paula here has asked about subtypes. What's a subtype, and how do you identify it? What's the difference between that and a variant?
I think as far as that's you're talking presumably about those three subtypes. Yeah, it's a very complex situation, because we tend to think of lineages, a human being being a hominid and going down, and when it actually branched off from an ape earlier on. Well, the same process happens with viruses, but a damn sight quicker. So there's a whole range of lineages and sublineages. And what happens is these all split off and have slightly different characteristics, not necessarily containing mutants that will affect the spike protein. So you could have a sublineage or a subtype that in fact is just slightly different in its composition, but very, very similar to to its cousins, if you like.
Thanks. Okay, I'm scanning this list. Peter Weston has got a question which I think we'll all be interested in. Peter's asked, do you think it's an advantage to have a different type of vaccine when we come to the autumn booster jabs?
Time will tell.
Yeah. I mean, there has been that. Sorry, go on, Jay.
I would say that with flu, we have different makes every year. Nobody takes any notice of it. So that may give us a clue.
I mean, we could end up in the future, not necessarily this autumn, but we could end up with the same situation where we have A, B, and C components present in the vaccine.
Yeah. Paula asked another question of interest to most people, I'm sure. Is a person with cold or flu more vulnerable to coronavirus infection?
Well, there is current evidence, it was sort of speculated to start with, that people who've had colds tend to have less vulnerability to coronavirus infection. Because there's some sort of cross reaction, basically.
You've already started the immune system on something.
And the immune system is already activated, and there's something called interferon, which is a cytokine that helps protect cells. So, and also your nose may be physically less able to breathe in another set of virus particles.
Well, it's really good to know there's a benefit in catching flu then.
But not at the same time as.
Right. Okay. Where are we, Barry? Have you got?
Well, let's take Mike Meadows has had his hand up, so let's take that, because he's Mike. Would you like to unmute and ask your question? No answer.
Or is that just a an applause hand.
It might be, yes. It does look like the hand up for question, but I'm hearing no answer. I'll assume that he hasn't. Oh, there's Mike. Can you unmute?
Yes, indeed. And thank you very much for both lectures. Always amazing quality and lots of information. The question I'd like to ask, which is a kind of elephant in the room, the present numbers of deaths are increasing. Who are these folks? What age are they? Ethnicity? And have they been immunized or not? We get these numbers appear in the media now, but there's no breakdown of what groups of people these are. Could you please comment?
I would totally agree with you, and I think I've been concerned about this for some time. I think there is evidence coming out. I mean, there have been some indications that most of the people going into hospital and getting a severe disease had either only recently been immunised and therefore haven't acquired full immunity, or they haven't been immunised at all. So I think that is the key message. But I agree with you entirely. I think sometimes there is a difficulty. I think there was a big concern about the BAME, the BAME sort of ethnic groupings, and the fact that they were getting more disease. I think what we can't forget is that there are a huge range of genetic predispositions, for example, to blood pressure, as one example, to diabetes, and to other inflammatory conditions, which occur more in some groups of individuals than others. And so, yes, there may have been an increase in some of those individuals, but there were reasons which we may not totally be aware of that could have contributed to that.
It is said that hospital admissions are still rolling along. They're not going up very much, but they are there. But they do tend, a lot of them seem to be younger. They're in hospital for a shorter amount of time. But with the numbers of the people that are being infected, you're bound to get some that will get into hospital. But I haven't seen any breakdown of the deaths, other than it's always mentioned that they're less likely to be vaccinated, fully vaccinated.
Less likely.
Can I just ask one question of my own, because it's intrigued me, and I certainly discussed it with others. Are there any prospects for an oral version of the vaccine?
Can I just come back on that, because I mentioned about plant-based vaccines, and there is the problem here is dose, isn't it? How do you get the right dose of vaccine in? But there is a prospect that you could get an oral vaccine that way, either through seeds or leaves, etc., of plants which are
plant-based.
And that it has been suggested that you could actually get an oral vaccine that way. I'm not saying there aren't other ways, but that's one. I mean, one of the problems is getting it through to an area where it can actually be taken up and to actually get to the immune cells, which there are lots of lining the gut. But you've got to get through the acid of the stomach and everything else first. So you'd have to put it into something like a liposome or something which would actually protect it on its way through the system to absorption.
Thank you.
Thank you, David. Any more questions from the floor, as it were? If you'd like to put your virtual hand up, that's great. Non from Cardiff. Yes. Would you go,
Hi.
please?
I hope I'm audible. Yes, I wanted to know about herd immunity, and I wondered if the ease of mutations that's been going on, or was going on, would affect the efficacy of herd immunity. In other words, would we get immune from all of the mutations, or would that interfere with those?
Interesting point.
Herd immunity is an interesting term. We are not going to eliminate the virus. We haven't been able to. Smallpox is the only virus we've ever eliminated from the human as a human host. Even something like measles, where we would say we had herd immunity. We only achieve that by vaccinating all the as many of the susceptibles, i.e. children, as every susceptible comes along. So it depends on vaccination is going to be the way that we deal with increasing our immunity. But at the moment, the vaccine is dealing with all the variants and mutations that come along. So we are hopeful that in the future, we will be able to deal with any variants. Do you see anything else, David, as far as vaccines go?
No, I think we tend to overlook the natural immunity that people who got the disease, the antibodies and the T cell responses that they've made. The overall immunity, it's been suggested at the moment, is something probably in the 90%, isn't it? Mm. Which for any virus normally is a pretty high number. I think, yeah.
It's astounding.
A remarkable number. So if we haven't got close to it by now, I can't see where, like you say, I can't see we're going to get any much better at it. We can't possibly eliminate every COVID virus. We can't predict who may or may not get it, even though they've been immunized. That's one of the problems we have.
Mm. We're not hearing very much in the press now about the R number. That used to be quite a dominant figure early on. Every day we'd hear what it was. Do you know what it is now? Is it relevant anymore?
It's about 1.
It's about 1. Okay. I read in the newspapers part of the reason that we seem to be on a downward slope in terms of hospital admissions and the infection rate is that we're just not meeting as many people as we did pre-pandemic. And we're rather slower than the scientists modelling suggested we would be in getting back to our pre-pandemic behaviour. Would you agree with that?
Well, I think again, the real world data suggests that there has been no spike in infections that is detectable yet after the opening up of
restrictions on the 19th. Yes.
So yeah.
Yeah. Okay.
Someone's just mentioned, followed up on my comment, smallpox. Smallpox was eliminated. I thought that's what I said. Smallpox is the only virus that has been eliminated from the human population.
Except in laboratories in Russia, England and America.
Well, not in England, not in England. Yeah.
Are young babies still being, or children being vaccinated against smallpox?
Oh no, vaccination stopped 20 years ago.
Did they really? I can't remember.
Yeah.
Yeah.
Okay. Yes, it was a wonderful campaign, and we've been trying to do that with polio ever since, and even with vaccination, we're not quite there yet with polio.
No. Got quite close, didn't it?
Mm. Afghanistan is the real problem.
Yeah.
Okay. Any more questions? Would you like to put your virtual hands up? No? Well, I think what a fantastic morning we've had with Jane and David. I'm sure everybody's learnt a lot. There's been plenty of opportunity for questions. I hope you've got the answers you've been looking for. There were a few other comments, more comments than questions, I think, on the chat box that we haven't really had time to run through. But I'd like to thank you, Jane and David, for a brilliant double act this morning, and please everybody join with me in giving them a round of applause. It was absolutely marvellous. Thank you.
Thank you very much indeed.